It is my great pleasure to introduce to you Dr. Vinod Shah. Dr. Shah is a pharmaceutical consultant, a member of steering committee of non-biological complex drugs, and expert consultant with NDA partners. Dr. Shah worked at FDA from 1975 to 2005, where he developed several regulatory guidances in the area of dissolution, SUPAC, bioanalytical method validation, topicals, bioequivalence, and biopharmaceuticals, and pioneered method development of IVRT for semi-solid drugs. Dr. Shah was the scientific secretary of International Pharmaceutical Federation and past president of AAPS and FIP. Dr. Shah.
Thank you, Kailas, for an introduction. First of all, I would like to thank Dr. Sam Branney and the organizers for inviting me today to make the presentation on the in vitro release test, the historical perspectives, current context, and the future directions with respect to the IVRT. My interest in the topical drugs dates back more than 5 decades when I was at UCSF before joining the FDA. After I joined the FDA in 1975, we found out that there was no suitable method for determining the drug release of bioequivalency of the topical drug products. For our research in the dermatological areas, we were always in touch with and trying to get the guidance from Dr. Howard Maybach and his group at UCSF. For our research in the dermatological areas, we were always in contact with Dr. Howard Maybach at UCSF and his group to get his guidance. Our joint first publication with Dr. Richard Guy and Howard Maybach was in 1986, where we talked about the bioavailability of dermatological and topical administered drugs. In this particular publication, it was pointed out very clearly that there are three sequential steps that take place after the drug is applied topically. The first step is the drug release from the dosage form. Second step is the drug penetration through the stratum corneum. And the third clear step is the pharmacological action. So with this as a background, can I have the next slide, please? The question came out is why do we do the in vitro release test? What is the genesis of doing this? We've noted that in 1987, Professor Richard Stoughton, a great dermatologist, published an article, Are generic formulations equivalent to trade name topical glucocorticoids? So when we see a publication of this, it's a great disappointment to the agency, and it is a disturbing factor that someone has published an article like this, which is really questioning the drug approval process of the FDA. Because of my interest in the topical drug products, I was asked to take a look into this, try to solve the problem, and come up with the solution, how to deal with these things. Knowing the fact that from oral drug products, the dissolution or the drug release plays a major role in the bioavailability of the dosage forms, we thought that a similar conclusion can be drawn as we saw it from the previous slide, that the first action after the topical drug application is the drug release. So maybe we should take a look into the drug release before we look into any other aspects. At that time, we also knew that many pharma industries and the drug researchers working in the dermatological areas were all familiar with the Franz vertical diffusion cell system using the cadaver skin. Therefore, we decided to develop the drug release method using the vertical diffusion cell and the synthetic membrane rather than using the cadaver skin, so that we don't need to worry about the reproducibility of the cadaver skin. Next slide, please. We decided to do our research work at the FDA lab in Texas. At that time, when I was at the FDA, there were several FDA laboratories across the United States, and all the labs dealing with the drug release or the dissolution, I was primarily responsible for that and trying to handle all the projects. So we decided to give this project at Texas. And we had our first brainstorm session with the late Bill Hansen of Hansen Research, the Crown Glass representative, I don't recall his name, but he was the one who was making the France diffusion cells for the Hansen research, the FDA chemist, Mr. Jerry Elkins, and myself. We all met in Texas, along with the equipment of the France diffusion cell, to discuss how exactly we should proceed and what are the different steps we need to take. And it took several times, several attempts for us to develop the first drug release method from the semi-solid dosage forms using the hydrocortisone cream. And that article was published in International Journal of Pharmaceutics in 1989. The research in the tropical areas then was carried out in several labs, in the FDA labs, with Professor Hans Schaeffer in France, with Dr. Gordon Flynn at University of Maryland, and Dr. Joel Zarch at University of Rutgers, and Dr. Lynn Pershing at University of Utah. Each of the investigators were looking into the different aspects of the drug release and the bioavailability, bioequivalency of the topical dosage forms. This was a new procedure, a new technique, and because of this, we decided to provide the training to the scientists, industrial scientists at the FDA lab. We gave this training on several occasions, 5 to 7 scientists, chemists at a time at the FDA lab, providing them the hands-on training so that they exactly know what is the procedure and how it should be done. After this, we had several symposia, meetings, workshops, all discussing with the IVRT. This was not an easy task. We had lots of opposition, the great resistance from the industry, as well as from the academia. I would be very clear to indicate that we did not have support other than those people with whom we are working in the lab with the FDA. The other academicians and the industry were not really in favor of this. We had a lot of resistance, but gradually we worked on it and so that it becomes acceptable to all. In about 1997, when the FDA guidance for the industry, SUPAC-SS, came out, that really also discussed the ways of comparing the two dosage forms using the IVRT, in vitro release. And that really helped to some extent for the people to realize it. Even in the training that we offered for the SUPAC-SS, we had also the discussions about IVRT. Later on, we had a workshop, FDA AAPS joint workshop, talking about the assessment and of the drug release of the semi-solid dosage form. And the report of that was published in 1999, and it really provided the usefulness of the in vitro release. Around this time in 1998, we had the first draft guidance on the topicals, which also stressed the importance and the value of the IVRT. Thus, IVRT slowly started gaining the acceptability. May I have the next slide, please? This slide provides a summary of the key publication. The first publication, as I mentioned earlier, was in 1989, where we talked about the drug release of hydrocortisone from the cream. The second one is the one with Professor Hans Schaeffer, where we really compared the pharmacokinetics, or the Demeter pharmacokinetics to be exact, the pharmacodynamics, and the drug release of the two dosage forms. And there was an excellent rank order relationship in all the three cases of the product. And that was published in 1996, I think. And then later on, we had the publication where we talked about the drug release and the skin branching of the beta-methazone valerate creams. that was published in JPharm Sci. We also had a publication with a Professor Zarts, where we talked about the release of the drug from the ointment. So all these individuals were tackling the different aspects of the drug release from the semi-solid dosage forms. Then we, as you know, we had the guidance of SUPAC SF 1997. During this period, we realized the variability associated with the in vitro release test methodology. And also we felt that, and as again, people are talking about it today, and we discussed yesterday also, that there is a need to standardize the procedure. If you don't have a standardized procedure, you cannot get the reproducible data that could be compared from one lab to the other lab. So we realized the variability of the dosage form and also the standardization procedure. To address these things, two collaborative studies were designed and were carried out using a reference product, a betamethasone cream, a betamethasone ointment, and a betamethasone dipropionate gel in 4 laboratories. After the first study was over, all the chemists were provided the training exactly how to do the things, what should be the precautions they need to be taking it, and the experiments were repeated. All these results were analyzed statistically, and it clearly proved and showed that there is a need to use a performance standard and suggested the training necessary for having a reproducible, reliable data. And that was published in 2007. This is all the background, some of the key publications. May I have the next slide, please? So now let's talk about where are we today. Today, as I see it, we already still have the SUPAC SS guidance, which was published in 1997, more than 2 decades. It is still in use, and it may be needing some minor tweaking here and there, but I think still see that it's a fairly good developed guidance. Recently, there are quite a few product-specific guidance publications, which has been published by the OJD and the FDA, and that refers to requiring the in vitro release testing requirements. In addition to the in vitro release, some of the guidances do require additional physicochemical testing, the rheology, etc. And in some cases, they also require the IVPT studies. Those are described in the product-specific guidances, and that's what's happening today. And I see that that's a very, very valuable things that has been undertaken by Dr. Sam Rani and his group to really develop the product-specific guidances. Still, there are some problems with respect to the methodology, the validations and all, and that's what we are discussing it in this particular workshop. And I'm hopeful that soon we will be coming to standardizations of all these aspects, and it would be quite useful. Today, the research is going on in the FDA labs, in academia, industry, CROs, etc. And all this, some of the major research is also funded by the FDA, and that's a very thought-provoking and trying to bring out the new data, new information on dealing with some of these issues. We also have today the USP Chapter 1724 that describes the IVRT methodology and also how exactly to carry out the drug release, the different equipments and all that. And that may be also undergoing some updates, but that's already in practice now. And so what this means is the IVRT, or the in vitro release methodology, has been well accepted now by the academicians, the regulators, and also the industry people. So that's a very good thing to know about. The other thing which I think it's very great, and I really welcome this, is the change in the requirements of the topical drug products Initially, the concern was to have the products Q1, Q2, and Q3. And now, probably as I notice is, the Q3 sameness requirement has been changed from same to similar. And I think that's a very welcome process, at least from my point of view, looking into the future of the topical drug products. May I have the next slide, please? So now let's talk about the future. What I see as the future might be there with respect to the in vitro release. First, I see that the research in the IVRT and the IVPT will continue towards reducing the regulatory burden for the bioequivalency requirements of the topical dermatological drug products. The research will be continuing at the FDA, with the academia, and also with the industry. I also see that there'll be a continuous publication of more and more of the product specific guidances, not only for the simple drug products, but also for the complex drug products. Though right now, according to the latest FDA definitions, we call all these topical drug products as the is a part of the complex drug products. So I see that there'll be more and more publications of the product specific guidances, which will be dealing with the IVRT, which will be making use of the IVRT. I also see that in future, there will be some in vitro bioequivalence approaches for the topical drug products. This is also one of the ways of approving the drug products, determining the bioequivalency. And I see that in vitro will be playing a major role as the time goes by. I would like to see that IVRT becomes a mandatory for semi-solid dosage forms, similar to the dissolution of the oral solid dosage forms. Also, I would say that there will be more and more applications of the IVRT during the R&D stages, in selection of the optimal candidates, as a quality control tool, as a stability, and also in the stability studies. In the future, I also see IVRT functioning as a tool for the biowaivers, especially in two cases. One would be the lower strength of the semi-solid dosage form, and second one, I would imagine that in future, the biowaivers would be feasible based upon the TCS, which is the topical drug classification systems, similar to the BCS. May I have the next and the last slide, please? So this slide really shows the comparison of the biopharmaceutics classification system and the topical drug classification system. I will not go into the details about the BCS, but I would just say that in the BCS, right now, if you see it, the drug products which fall into the category of class 1 and class 3 are eligible for the biowaivers, whereas the drug products will fall into the category class B and class 4 requires the bioequivalency studies as far as today's BCS guidance and the regulatory requirements are concerned. Now, let me briefly talk about the topical drug products classification system, TCS. This is a fairly new one. The TCS is a framework for classifying the topical drug products based on the Q1 and Q2, based upon the role of inactive ingredients, based upon the microstructure and the in vitro release. In our hands, we have found that in most of the cases, as we make the changes in the formulation or in the product, there are changes in the microstructure, but which are also reflected in the in vitro release of the dosage form. So let's talk about the class 1. In the class 1, the Q1 and Q2 is the same as the reference listed drug, and it reflects the level 1 changes in the SUPAC-SS guidance. In the case of class 3, the Q1 and Q2 are different than the reference listed drug, but have the same functionally similar inactive ingredients that results in the Q3 similar to RLD. Again, Q1 and Q2 are different, but functionally they are similar to what is in the RLD, and that's why it results in the Q3 similarity. This is in line with the current thinking of the evolving concept of Q3 same to Q3 similar. According to the TCS proposal, the generic topical drug products falling into the category 1 and 3 will have a similar release profile and will be eligible for the biowaiver. Class 2, where Q1 and Q3 are similar to a reference listed drug, but have the different Q3 and therefore also have the different in vitro release. In the case of class 4, all the aspects, the Q1, Q2, Q3 are different, and IVR is also different. And the generic products falling into these categories will be required to do the bioequivalence studies as per the agency requirements. We are currently continuing our research at University of Romania towards further validation of the TCS concept. In conclusion, the in vitro relief test has come a long way. It has established its position in the drug development and evaluations of the topical drug products. Next slide, please. Thank you for your attention.